I have the privilege of introducing Dr. Keith Delaplane today. Keith is a professor from the University of Georgia, the Department of Entomology. Did his Bachelor of Science at Purdue in the entomology department, right? Keith,
Animal science.
Oh, animal science. Oh,
Here's
okay.
a story for you.
Okay. And then a PhD at LSU with John Harbaugh. Keith, right now in his position, has a two-way split between research and extension. So done some fun work on pollination, Varroa IPM, and polyandry, which I believe he's talking on today. Another fun fact, Keith has an MBE. He's a member of I'm going to get this wrong, a member of the British, Keith, what is the full acronym? I can't remember.
Yes.
There we go, Empire. Ben Taylor is shaking his head at me right now, my postdoc in London. Okay, Keith, take over.
All right, Brock, thank you for that introduction. And thank you for the opportunity to speak for your department. Thank you for zooming in with me here today and in fact, accommodating Zoom. And I'm happy to talk to you about this subject because it bridges all aspects of my appointment in extension and research. It has a great deal of information about how social evolution unfolds, by implication, how many other transitions in evolution unfold, and it has practical application for our client groups with beekeepers. So with that ambition in mind, I'm going to talk for the next 45 minutes about multiple mating in the honeybee queen and its implications for social evolution and honeybee health. At the risk of overgeneralization, a repeating pattern in the history of life on planet Earth is the subsummation of lower independent replicators into larger groups that then collectively pool their genetic fates together, reconcile genetic conflicts, and engage in high cooperation to achieve greater ecological fitness. So the necessary requisites for a true individual in evolutionary speaking is high degree of cooperation and the resolution of those earlier levels of genetic conflicts. And every time this happens, we consider this the emergence of a new level of individuality in the history of evolutionary transitions. This has been an idea developed since at least 1995 with Maynard Smith and Smesmari. And it's the idea of these lower replicators lumping together. And I've just illustrated this at the bottom. We have unlinked replicating nucleotides that then join together into replicating nucleic acids. In turn, we have nucleic acids that join together into bundles bound in cells as chromosomes. Then we have cells that bundle together as multicellular organisms. And it's at this stratum, of course, where insects and where you and I and plants exist. The next stratum above that we colloquially call the superorganism because this is the subsummation of units that are themselves organisms. Think of it as a honeybee colony. The individual workers are already organisms, multicellular organisms, but they have come together in a high degree of cooperation and they have resolved interior genetic conflicts so that now the group is operating as a unit of selection. Once you have those criteria, former replicators coming together into a high degree of cooperation, they resolve their interior genetic conflicts, and ecologically, the forces of selection are greater between groups than it is within groups. We can now formally recognize this level as an individual. Individuality is a term in the broad spectrum of evolutionary history. The superorganism, you can think of it by other words that may be more familiar to most of us. Eusociality, eusocial species, social insects that are not just social, they are eusocial, truly social. And these are criteria that go back to 1971. E.O. Wilson, in his classic book, Insect Societies in 1971, said that for a species to be eusocial, it has to meet these three criteria. It expresses cooperative brood care, reproductive division of labor, and overlapping generations. But even within the level of a superorganism, we still have hierarchy. We have what we call the simple eusocial species versus the complex eusocial species. And there are some qualitative and quantitative differences between these two. And I've sort of outlined a few of them here, using Hymenoptera as an example. Simple eusocial societies have workers and reproductives that are essentially indistinguishable from one another. A good example of this is the bumblebees. Bumblebee workers have tremendous variation depending on their quality of diets they receive as immatures. And so you can have workers that are along a gradient of kind of very, very worker-like, all the way up to larger and more queen-like, until by the end of the season, a bumblebee colony is basically producing reproductives. But morphologically, there's no distinction between what we would call a worker or a reproductive in bumblebees. That distinction is quite strong and quite clear, however, in the complex eusocial species, such as the honeybee, where the queens and workers are morphologically distinct. In simple eusocial societies, the workers are reversible. They can return to, and often do, return to reproductive forms. The workers will mate and produce brood, whereas the castes are irreversibly fixed in the complex insect societies. And you do not have this ability to be plastic and go back to an earlier state of of a working capacity, so to speak, or reproductive capacity if you are a worker. As a consequence, there's much more variety of castes in the complex eusocial societies. You not only have castes that are different in worker function versus reproductive function, but within the workers, you will have specialists. In the termites and ants, for instance, we have major workers, minor workers, medium workers. We have workers that are adapted for defense, extremely large mandibles, phragmatic blocky heads to block tunnels, for example. We have many caste forms of morphological diversity, and we only find this in the complex eusocial societies. Simple eusocial societies can retreat to a solitary state, and we see this again in the bumblebees. We see this in Polistes wasps, most wasps, in fact, where toward the end of a season, the colony will produce a reproductive, the reproductives will mate, and all of the workers and the males die, and it's only the recently inseminated queens who then go into a winter hibernation to start their life cycle up again the next spring. The complex eusocial societies do not do this. They are committed to year-round eusociality. They do not lapse back into a solitary phase. This, of course, is the case with the honeybee. As a sum of all this, in simple societies, we have colony populations that are relatively small. In the halictids, for example, we have some colonies which number just a few individuals, even less than 20. In bumblebee colonies, we usually have individuals with number in the tens, but it's in the complex eusocial societies that we find the truly large populations, honeybees with populations of the tens of thousands, 50 to 60,000, ants and termites, which can exceed 1 million members. And these levels of complexity are only seen in the complex eusocial species. And then finally, and near and dear to my heart, and the subject for the rest of this presentation, in the simple eusocial societies, monandry is common, whereas polyandry is common, or its equivalent, functionally, polygyny, multiple females that are reproducing in ants. But since we're talking about honeybees, honeybees only practice polyandry, mating with multiple males. This is common in the complex societies, whereas the simple societies tend to have monandry. The effect of this is colonies whose members are very genetically diverse. And polyandry and increasing degrees of polyandry has been traced for at least 15, 20 years now to many measures of colony fitness. It is patently a colony level adaptation, because once in a hymenopteran colony, the mother begins practicing multiple mating, the average relatedness of her daughters plunges considerably. So there is a real cost to the daughters once their mothers begin practicing polyandry. So polyandry can be understood best as a colony level adaptation, not an individual level adaptation. Altruism plays into this. How do individuals abandon selfish interests and pursue altruistically the reproductive fortunes of another individual has been a a mystery all the way back to the grand old man himself. Charles Darwin was not privileged with knowledge of Mendelian genetics, even though he and Gregor Mendel presumably were contemporaries. There's no record that they actually knew each other and were familiar with each other's work. If Darwin had been familiar with Mendel's work, he probably would have made a qualitative leap forward in understanding how altruism can evolve in insect societies. As it was, he still hit pretty close to the mark. He said that altruism could evolve, as he called them, the neuters. He says altruism could evolve in the neuters of insect societies if there is enough benefit to near kin to justify the altruistic behavior of the altruist. So he was hitting terribly close to the modern concept of group selection. But as it is, it took about 150 years for us to get the mathematical explanation of this with the work with Bill Hamilton in 1960s, who gave us so-called Hamilton's rule that shows how altruism can in fact evolve in a social insect society or social society period if the cost of the altruistic behavior to the altruist is less than the benefit to the recipient multiplied by their shared relatedness. And John Haldane came after Hamilton and put it this way, which is poetic and mathematically correct: I would lay down my life for 2 brothers or 8 cousins. So group selection has pretty much owned social insect evolution since Hamilton's work. Kin selection has been elevated and called by some as a general theory of evolution, and it's been very robust against many challenges in recent years. In the case of the Hymenoptera, Hamilton is rather on steroids, we might say, because of Hymenopteran rules of inheritance. Individual sisters are not related by a simple 50-50%. They are related at a level of 75%. So when it comes to Hymenoptera, Hamilton seems to knock it out of the park. If there was ever a case for kin selection, it would be in the Hymenoptera, right, where the sisters have such high degrees of kinship that it made understanding the evolution of altruism almost compelling. Well, of course, it's not quite that simple, is it? Because honeybees and others that are practicing complex eusociality don't just mate once. Hamilton's rule applies only to super sisters, sisters who share a common father. For an assembly of workers of mixed paternity, Hamilton no longer counts because the average relatedness drops precipitously with a colony whose mother has multiply mated with many males and stored their sperm and produced a cohort of workers with mixed paternity. For these individuals, average paternity is more on the scale of 30%. So the workers clearly take a hit simply looking at value of reproductive value to the next generation. They take a hit. The queens are still propagating 50% of their genes with every reproductive event, but her sterile daughters have really taken a hit. So it can only be understood as an advantage at the colony level. So polyandry has been a winning strategy for the complex eusocial colonies. In fact, it is probably a cause and effect of their complexity. Polyandry was integral to the evolution of complex eusociality. It is not accidental to the evolution of complex eusociality. It is only in the complex eusocial species that mate multiply that we find the divergent behaviors among their workers that we celebrate so much in honeybees. I tell people that virtually everything you've ever heard about honeybees, it's the workers that do it. It's the workers who have the repertoire of behaviors, the ability, high cognitive skills to interpret symbolic language, to recruit, to make collective group decisions. It's in these societies that we find the most advanced and celebrated behaviors in the honeybee and other social species. So there's been a great deal of interest in what drives multiple mating. It was certainly a barrier to be overcome in early social evolution. It's probably no accident that complex eusociality has only evolved maybe I think the latest number is about 26 times in the history of Earth. Only 26 times has there been an independent evolution of complex eusociality. So the genetic barriers are formidable to overcome. To remember those criteria for the emergence of an individual, you had to have good degree of cooperation, but you also had to have a resolution of genetic conflict. And so how is this done in a species that begins practicing multiple mating when by definition their kinship has dropped precipitously? Well, we're going to get to that in a minute. But let's take a look at some of the math behind what's expected in a Hymenopteran family. What you're seeing here is a plot of the average within nest genetic diversity, the percentage of shared alleles, average number of shared alleles between individuals, relative to the x-axis, which shows the number of matings of that colony's mother. And you can see how it plateaus in the Hymenoptera at a level no higher than 75%. At about 10%, that line begins precipitously bending flat, which means on average, a queen has captured about 90% or more of the alleles in her breeding population by her 10th mating. Nevertheless, the species continues mating beyond this asymptote. In fact, the species average for Apis mellifera is about 12 males. Yet we have in the literature records up to 44 males, and the highest, to my knowledge, is one record of 77 males in a naturally inseminated queen. Which begs the question, what are the adaptive benefits of polyandry level at such exceeding excessive levels? If they've already exhausted mechanistically most of the alleles in the breeding population, what drives them to continue these supernumerary matings? Mating behavior is not selectively neutral. There's risks involved. There are ecological risks. There's predation. Queens are not necessarily very good flyers, so they're vulnerable to bird predation. They're vulnerable to sexually transmitted diseases. So one would think that this would cap earlier than this if we were simply looking at genetic diversity as the driver. Well, there's been some hypotheses put out there to explain these supernumerary levels of polyandry, which I call hyperpolyandry. I call hyperpolyandry anything that's beyond that predicted asymptote. Anything beyond that, which is common in honeybees and in the army ants, anything beyond that, I'm calling hyperpolyandry here. Well, some explanations is it could reduce sex ratio conflict. Remember, one of the criteria to emerge an individual, you got to resolve these reproductive conflicts within the group. And if I am a Hymenopteran and my sister has 75% of my genes in common, I'm going to want to reproduce more sisters. Whereas my mother, who transmits 50% of her genome with every egg that she lays, she has no such motive. Her queens will be adapted to favor a 50-50 sex ratio in their offspring, whereas her super sister daughters will be exactly the 3-to-1 ratio. They are predicted to prefer rearing more sisters at a ratio of 3-to-1. So if the mother practices polyandry, she has immediately dialed down the conflict for this sex ratio contest. Otherwise, you'd have a bunch of subfamilies that are vying to produce their own super sisters as the colony's next queen. But if there's a large number of patriline's under that roof, it diffuses that competition among the patriline's and hopefully improves social cohesion in the group. Another explanation for hyperpolyandry is it reduces inbreeding at the sex locus. Sex determination in Hymenoptera is established at a locus just like any other character. The numbers of alleles for this are in the neighborhood of 20. And with inbreeding, you can get homozygosity at these loci and cause sterile males. And this is very maladaptive at the level of a colony. So mating with multiple males reduces the possibility of happening this. Ecologically, a colony whose mother has mated with many males should be expressing a lot of phenotypic averageness with other colonies within its population. evolution is not necessarily directional. Often evolution is static. It is adaptive to be average. You don't necessarily want to be the tallest sunflower head in the field, or certainly not the lowest, because you'll be shaded out. Average is what's rewarded. And by practicing polyandry, a mother is capturing more and more and more with every mating, all of the alleles in her breeding population, and her colony will be less prone to phenotypic extremes. Polyandry has been shown to increase robustness against parasites and pathogens, presumably through the genetic diversity of the patriline's, which in turn have more specialized behaviors for resisting the parasites that threaten them, which is tied into the last bullet there, improving task efficiency through specialization. Social colonies that have a lot of specialists minimizes the necessity for any one individual to perform a series of tasks serially. Specialists allow one task to be performed simultaneously with an improvement in overall efficiency on ecologic fitness of the colony. We're going to be coming back to some of these criteria because there are more when it comes to real extreme hyperpolyandry. With the technology of instrumental insemination, we can test some of these ideas and look for measurable effects on colony fitness from queens who've been mated with multiple males. And your colleague in your department, Crispin Given, helped me with this project. We got this published in, that's the next paper coming up. This one Crispin wasn't with, sorry. This is a project that I did when I was on sabbatical in England with my colleague Giles Budge. And we were interested in looking at colony level benefits to hyperpolyandrous matings. Now notice that all of these numbers that we looked at are above that genetic diversity asymptote. They were already higher than the genetic variance model would predict. So we were kind of interested in whether colony fitness kept improving above that genetic diversity asymptote. So we were inseminating queens with either 15, 30, or 60 males. These males were not selected for any character. They were just simply identified from different colonies we had in the apiary. There was no genetic selection on our part happening, just wild type drones. And we were able to show an increase in brood rearing efficiency when we jumped from 15 drones to 30 or more with our insemination number in our queens. This effect, by the way, in my opinion, is one of the most robust benefits of polyandry. We keep seeing benefits to brood rearing efficiency, brood cells reared per 100 bees, brood survivorship when it's still in the larval stage. This is a benefit that seems pretty robust across a lot of papers published on polyandry. Exciting to us, this was the first paper to our knowledge that showed a benefit of polyandry with the parasitic varroa mite. And any of you following honeybee health and pollinator health, you probably know about this parasite. It's an exotic mite parasite of Asiatic honeybees that has been spread around the world. And when it comes to parasites, it's generally considered public enemy number one for managed honeybees. Our Western honeybee, Apis mellifera, has relatively few genetic resources to wage against this parasite, and it's been a very dangerous parasite as well as a vector of many serious honeybee diseases. We were able to show that at polyandry levels in excess of 15, we had fewer numbers of colonies testing positive for this mite. And so this is exciting, and we have since found more evidence to back this up. So beekeepers love this kind of stuff because it shows very clear economic benefit as well as colony level fitness for evolutionary biologists. It's a pretty cool area to be working in. Back to this idea of hyperpolyandry and why, okay, we know all of those hypotheses that I mentioned in the previous slide apply to all of polyandry, all the way up to the asymptote and beyond. But what compels a species to go so much far beyond that? We were interested and alerted to this paper, a theoretical work by Stefan Fuchs and Robin Moritz. It was published back in 1998 or 9. There's variable publication dates on this paper. But they put forward some hypotheses I think make sense for hyperpolyandrous mating. How could this evolve? And these are the final 3 bullets there that Fuchs and Moritz put forward for hyperpolyandry. They say it can increase acquisition of rare specialist alleles. Sure. Many of these very specialized characters at the colony level are in fact rare. I'll be talking about one in a minute. If we had, for example, colonies that were entirely composed of specialists, by definition, we mean that we have relatively fewer generalists. And we still need generalists to do the important stuff. We still need generalists to forage, to ventilate the nest, to do everything that's necessary for survival. For rare parasites, we need fairly rare resistance alleles. So these specialists very often tend to be either recessive or just simply rare in a breeding population. And if a queen goes beyond that genetic asymptote of flatline, it increases her chance of capturing those capture those brass rings, if you will. Fuchs and Moritz also put forward the possibility that it may not simply be the raw acquisition of alleles that matters. It could be the acquisition of new and unique allele compositions, genotypic compositions. And they point out the obvious, that though the alleles are finite, the allele compositions are technically infinite. And by practicing polyandry, a queen could be continuing to come up with new allelic compositions among her patrilions that optimize colony fitness. Finally, they put forward a third bullet, a third hypothesis, somewhat tied to the previous 2, that by getting these specialized alleles, or by getting these specialized and unique genetic compositions, that a colony could avoid cliff edge catastrophes, which is a term you'll find in the ecologic literature. If you have a rare allele that's a specialist in a population that really confers high survivability to its possessors, a member of that population could fall off the cliff, so to speak, if they fail to capture that specialist allele. Hyperpolyandry can be seen as an extravagant attempt to avoid such catastrophes. And these were what seemed to us testable hypotheses in the latest paper that we just got out last year. And this is the paper which your colleague Crispin cooperated with. Crispin performed the instrumental inseminations for this study that I'm going to be telling you about now. We were looking for a candidate allele that would fit some of these criteria of being rare with extreme colony level fitness benefits. And the one we came up with is so-called hygienic behavior in honeybees. This, we call it hygienic because it's an expression of workers being able to identify brood that is compromised. If a brood cell is positive for pathogenic fungi or pathogenic American foul brood disease, they can detect these compromised cells and open up their contents and remove the prepupa or the pupa along with the parasite and disrupt the parasite's life cycle. It doesn't necessarily kill the parasite, in this case varroa mites, but it certainly interrupts their reproduction. And Marla Spivak at the University of Minnesota has championed this line, and she has ably shown that it has colony level effects of reducing varroa mite numbers. Hygienic behavior can be identified in the field by proxy if the investigator or the bee breeder uses freeze-killed brood. And I'm showing you this thumbnail here of a PVC pipe or a canister that can be screwed into a comb of sealed brood. Liquid nitrogen can be poured in, which very quickly freezes the brood rock solid. And then you return that freeze-killed brood to the colonies, and colony at the bottom right has removed only a modest amount of the freeze-killed brood, whereas the colony on the bottom left of that thumbnail has removed all of that freeze-killed brood, and we would expect the colony on the left to be expressing complete hygienic behavior. Hygienic behavior is in fact rare. It's only about 9 alleles that control for it, and it has measurable and demonstrable colony fitness benefits. So this was our model rare allele in our design, using instrumental insemination to make a factorial design between the presence or absence of varroa sensitive hygiene drones and the presence or absence of low or high polyandry. Here is a a mock-up of our factorial design. We tested polyandry inseminated queens with either low 9 males or 54. And we inseminated them accordingly with either VSH drones or wild type drones. Why 9 and why 54? It's simply multiples of the numbers of our drone source colonies that survived over winter. No more reason than that. But it also conveniently brackets that qualitative divide between the flat line that asymptotes at about the 10th mating versus that level of mating wildly beyond that asymptote. So we were trying to capture qualitatively polyandry within that GV asymptote and polyandry above it, and try to detect a colony level fitness benefit for actually practicing hyperpolyandry. Brood survival. What you're seeing here is a whisker chart showing how what we're measuring here is the 3-day survivorship of brood. You can go into a colony, mark open brood that's still being fed and tended by nurses, and come back after 3 days and see if those same brood are still alive. It's quite simple to do. And this was a significant difference in distributions. On the left, we have lower brood survival than on the right, where we have the colonies whose queens were inseminated with 54 males. If you try to figure out the means on these boxes, you're looking at a difference of about 84% versus mid 90s, and which sounds small, but an average survival of a colony of 84 versus 95% survival can be ecologically quite significant. I'm going to show you a demonstration of that here in a minute. We were also interested in varroa mite levels amongst these 4 treatment combinations. And we were able to show a significant drop in varroa mites, but in VSH colonies, but only in the VSH colonies whose queens were inseminated with 54 males. It only expressed in the 54 male colonies. There was no difference between mite levels with colonies across the board with polyandry at levels of 9. We were trying to put this into context. With varroa sensitive hygiene, other papers have shown that it does not operate when it is only present at low rates in a colony. Remember, I was talking earlier, Fuchs and Moritz hypothesis said they have to go out there to get those rare alleles. VSH happens to be one of those resistance characteristics that has to be present in very high levels in the colony for it to express. And now this is based on a paper that Marla's group published with American foul brood as a model disorder, not varroa mites. But if that is true with American foul brood, then we're assuming it's true with varroa mites, that there has to be a large number of workers in a colony carrying the VSH and expressing the VSH character for the colony to have a reduction in mite levels. So we modified that original Fuchs and Moritz hypothesis a little bit to say that for those characters that are rare but have to be expressed in a colony at a high rate of individuals, hyperpolyandry is helpful for that too. So this could be an additional hypothesis for why hyperpolyandry could evolve if there are specialist characters that do in fact need to be present in a colony at very high rates. Very often the case is the opposite. You can get beneficial effects on a colony when there's only a few numbers of individuals carrying the specialist trait. These individuals tend to be keystone characters. They're influencers that have very strong impact on their sisters in the nest. VSH does not appear to be one of those. We simply need to have a lot of workers expressing VSH for it to show colony benefit. So this has been shown with AFB, American foul brood disease, and we believe that our results are consistent with that interpretation when the specialist character has to be available at high rates in the colony. Okay, all of the sum effect of this was in those colonies in which the VSH queens were present, by the end of the season in November, we had significantly greater bee populations. I'm focusing on November in particular. The blue line are those colonies with VSH drones, regardless of whether 9 males or 54 males. If they simply expressed VSH, their survivorship was better at the end of the year. Apparently, the benefit of that interaction, the VSH at 54, was big enough to pull the entire average for VSH colonies in favor of survival by November. So it was a pretty big effect size of those VSH colonies inseminated with 54 males. Take a look at the populations here. I was talking earlier how small changes in survivorship can have big effects. We're talking there an average distance. I'm eyeballing my graph there, about 3,000 workers in the colony versus 2,000. This has pretty big effects on a colony's overwintering survival when it comes to November. So small changes in survivorship of individuals can have big effects on fitness of their colonies. I want to move on now to a new paper that we're just getting ready to submit. When I'm speaking to beekeeper groups, they like these kind of results. Multiple mating is good. Multiple mating improves brood production. Multiple mating improves varroa mite resistance and a host of other characters, including foraging efficiency, resistance to chalk brood disease, thermoregulation. There's a lot of good economic characters as well as ecologic characters that improve when a colony has a lot of genetic diversity. So how can we translate this to beekeepers? The results I've shown you so far are entirely dependent on instrumental insemination, and beekeepers, as a rule, it's been hard to translate that technology in any meaningful scale to the beekeeping industry. So we were thought, what are alternative ways we can deliver the benefits of polyandry that beekeepers can use? We did a design here, another factorial design, where we were interested in the effects of polyandry interacting with a pesticide challenge with a neonicotinoid. And we did this by mixing brood. Those of you who are familiar with honeybees and beekeeping know that our standard equipment is interchangeable. Combs can be moved liberally from one colony to another. So it's possible to take brood from one colony and transfer it to another. In fact, it's common. Beekeepers do this a lot as a part of normal spring manipulations. So what I've shown you here are 3 colonies of 3 frames of brood. And if we use an average of 10, we can say that each one of these colonies, we expect to have a paternity count of 10. Her mother mated with 10 males, let's say. So 10, 10, and 10. Well, what if you just simply redistributed the brood of those 3 among the 3 colonies like this? And you could conceptually now have simulated a polyandry level of 30 in these mixed broods. Now, granted, this state of polyandry is temporary. This cohort of polyandrous workers will live their life and die out by attrition. So it's a wave of polyandry, if you will. But if that wave could be timed such that it is to the optimum time of year for the beekeeper, whether that's a honey flow, to maximize incoming honey, or if it is at a time of year when varroa mites are geometrically growing in the colonies, if we could intercept that geometric growth curve with a benefit of pipe polyandry, as I'm demonstrating here, it could be economically important to them. We, with that factorial design, we used pollen supplement patties as the vehicle for delivering the neonicotinoid at small field relevant levels relative to the literature. And then we tracked these colonies for measure of fitness. So there you have it again. It's another factorial type of design, 2 levels of polyandry, 2 levels of a pesticide challenge, and we tracked these colonies for fitness. We found, again, a significant effect of brood survival. The clusters there of interest are the blue one and the purple one. Both of these are the groups of colonies in which the brood was mixed, and we caused a simulated effect of hyperpolyandry, and this effect was significant. there is significantly higher brood survival in those colonies which we increased polyandry by mixing the brood. Unfortunately, we found no benefit on mites in this study across the 4 treatments, and we also found no effect on neonic relief. The neonicotinoids, there was no difference in colony survivorship with or without neonics and with or without mixing. We were hoping to be able to demonstrate a rescue effect, and that was not apparent with brood mixing, which is a bit of a downer because this would be a very easy and deliverable technology to beekeepers who are facing increasing effects of pesticide exposure to our pollinators. Honeybees, of course, are point source foragers. They sample the habitat over a scale of square miles and return to their nest with the pollen from all their diverse sources. And their colonies are very rich gold mines of virtually all pesticide residues available to them in that large foraging range. So we know, we have evidence that there's a great deal of chronic toxicity going on with bees because of these low levels of multiple pesticide residues in their brood. It would be nice if we could deliver a remedial to this state of affairs, but we have no evidence yet that brood mixing or polyandry in general is that remedial. So I want to begin wrapping up here and kind of leave you with the ideas that I think are interesting, the ideas that I think are important. I remind you that natural history is a record of formerly independent replicators coming together and now reproducing as part of a larger unit. And when this happens, and when the group that results has a high degree of cooperation, and if it has resolved its genetic conflicts, and when the intergroup selection exceeds the selection inside the group, we say a new individual has emerged in evolutionary transitions. These criteria of high cooperation and a reduction of genetic conflict appear to be general, not particular to the social insects, and they can be applied up and down Smith and Chatenberry's evolutionary transitions. And they are exemplified in the social insects. I take pride in realizing that it's the social insect biologists who have been laboring over the decades who began piecing together these principles of group formation and conflict resolution that we are now realizing are general. They appear to be general patterns, that group formation across evolutionary transitions is essentially a social process. And it's the social insects that have sort of pointed the way and illustrated this from an early stage. So this literature is very rich in references to entomology and social insects in particular. Complex eusociality, as opposed to just simple eusociality, it's the most extreme example of the tension between cooperation and conflict resolution. It appears to be very rare. Only 26 times in Earth's history has species tipped over into complex eusociality. It's a hard sell to abdicate the individual's genetic interests for the good of the group, especially in a hymenopteran sense, when I am helping my mother produce more of my sisters with whom I am related by 75%. This was apparently practiced gradualistic or accomplished gradualistically by mothers by expressing polyandry, increased the colony level ecologic fitness of their colonies and dampened down the sex ratio conflict among their daughters. And when that damping down process was sufficient to improve selection between groups, then polyandry was able to take off and evolve the resulting complexities and wonders that we see in the complex eusocial species, the ants and all their huge colonies and ecologic preeminence, the ants and with all of their complex multiple castes, honeybees with their complex symbolic dance language. We see all of these marvels that we as biologists celebrate and talk about in the complex eusocial societies. Genetic diversity was integral to this, not accidental to this. We have shown that benefits from polyandry can occur at levels beyond what is predicted by simple gene capture. A mating mother has captured 90% of her alleles after her 10th mating, but frequently in the honeybees and the army ants goes wildly beyond that number. And we have been able to show colony level fitness benefits at mating levels of their mothers beyond that asymptote. Some of the best explanations for this are the capturing of rare beneficial alleles, capturing rare and unique genotypic compositions, all of which accrue to ecologic benefits. Resisting cliff edge catastrophes was the term I used. In our experiments that I've shown you this morning, a rare specialist trait only expressed at the highest levels of polyandry studied. And this is consistent with the idea that in some specialized characters, we need a lot of specialists in the colony to achieve colony level benefit. And we conclude by showing at least one proxy of hyperpolyandry that's pretty easy to translate to beekeepers. You can mix brood in colonies, and we have shown that this does simulate polyandry and the benefit of improved brood survival, which has very direct ecologic and economic benefits for beekeepers and the honeybees themselves. This is overall, in my opinion, a fairly understudied area of work when it comes to honeybee health. It is gaining a pretty robust literature in evolutionary biology, and it's being increasingly understood that polyandry is integral to the evolution of complex eusociality. But there's not been a lot of meaningful translation of this yet to our modern problems with honeybee health. And I think this is a ripe area of work. I know Brock is interested in this. A lot of labs are interested in this. It's an enormous gold mine for its basic biology and for its applied benefits. I want to recognize my many co-workers from Jeff Williams and Selena Bruckner at Auburn, Debbie Delaney and John Menz at Delaware, Barry Brosey, formerly at Emory, now at Washington out in Seattle, and Lewis Bartlett, my postdoc, Jennifer Berry, my technician, and my students along the way. And our funders for this work I've given you this morning. And with that, that's the end of my prepared comments. And I'm open for any questions or comments you may have. Thank you for your attention.
Thanks, Keith. That was really fun. A lot of really exciting experiments coming through. I think we have time for the 3 questions that are on here. So I'll just start with those right at the top. Okay, so this is from Abdel Holloway. The benefits of polyandry seem to mostly occur on the colony level. Not to be controversial, but is there an argument for group selection to be made here?
It's I very much agree there's an argument for group selection here. In fact, I would put an exclamation point on that. In fact, it can really only be understood at the level of group selection, in my opinion, because it's definitely not adaptive for the workers themselves. It really can only be understood in that context. So yes, I agree fully.
Could I add a complexity? And I shouldn't do this. I'm going to. It kind of depends on how we define an individual. If your individual is defined at the superorganismal level, then this is just selection, right?
Yes, yes, it is. But I do think at this level of the superorganism, that's almost the inescapable level that we need to discuss it at, because the individuals are no longer totipotent. They cannot go back and forth. A queen cannot do worker duty. A worker cannot do queen-like duty. So they are mutually dependent, and it's really only as a unit can we understand them ecologically. So I mean, I leave open, and you and I were talking this morning, Brock, about the idea of competition with patrolines, for example, or sperm competition happening inside the colony. It seems for it to be meaningful with this species, it would have to eventually express at the colony level. So I do think that let's put it this way. In my opinion, I think group selection is more important than individual selection with this species, with this level of social transition, evolutionary transition.
Okay. The second question is a clarification, I think, on hygienic behavior. If the hygienic alleles need to be present at higher frequencies within the colony in order to be effective, shouldn't those alleles be less effective in the high polyandry scenario?
depends on where on that continuum would we begin seeing the effect take place. And I assume that there is a trade-off going on there with, if we're getting more and more specialists, does that mean we're having fewer and fewer generalists? I think that's a fair question. And we could see a point of transition or optimization here where a cost to the generalist tasks is being exceeded by the benefit of the specialist's tasks. It's a brilliant question, and that's the kind of experiments that we would have to run to answer it.
Okay, thanks. And then one more here from Matt Dittman. Do drones exhibit reproductive competition with each other? So are all sperm packages capable of degrading their competitor's sperm?
Yeah, well, there is some evidence of perhaps behavioral competition. There's also evidence of behavioral cooperation. There's a fascinating area of work and ideas put out that the drones actually facilitate one another in multiply mating with the same queen. And if any of you who've worked with drone physiology or done instrumental insemination, you know that the endophallus of the drone, when it's everted, has very bright orange coloration. And there's been some work out of Poland that's shown that this orange coloration is actually attractive to subsequent drones, that it improves their capacity to find and mate in succession with that queen. So behaviorally, there's good evidence for cooperation among drones. Now, at the level of the sperm, it's certainly arguable, because one of the most common things we see is gross disparities in paternity expression. Just because a queen mates with 10 males by no means means that every worker has 1/10 of a chance being expressed. We find some paternities who have 75% of the workers sired in a colony are one male. So there's definitely some kind of competition going on post-copulatory with the sperm. And in fact, drone competition sperm competition is a very viable argument for an alternative explanation for hyperpolyandry.
Great, thanks. We're over time, but I see one more grad student question, so I want to make sure we get that one. This is from Garrett Slater. Oh, hey, Garrett. There are many benefits of extreme polyandry, yet the harmonic mating mean of US honeybee populations is only about 14 queens per mate. Why isn't the mating number higher? Does the benefit of polyandry diminish after 14 to 20 mates per queen? You can read that in the chat if I didn't say that correctly.
I do think, I think the implication of your question is sound, and the implication is, can we have confounding happen? And we do know in some complex systems, like this is a field in information theory, that you can have too many actors. You can have, colloquially, too many cooks spoil the soup, where you have different patrolines and specialists who actually confound and interrupt one another. I do think those dynamics are real. I do think they're out there, and they could be a limiter on the expression of hyperpolyandry. I do think there's a ceiling to this. Just what would it take to detect it would be formidable, practically speaking.
Yeah, I'm wondering if we should be measuring the number of drones or the relatedness of those drones that a queen is mating with. Is absolute number what matters? I don't think so. I think it's more the relatedness among drones that matters. But yeah, I guess so. By that prediction, I think it would, our differences in mating among populations might be dictated by average relatedness in a population. But I can't wait to have you come to a lab meeting so we can further discuss all this.
Me too.
I don't see any more questions coming through. Thank you everyone for attending. Thank you so much, Keith, for coming today. It was a pleasure to host you, even though it was virtual, but we'll get you back here in person one day.
Good. I hope so. Well, thank you all for listening. I appreciate it.
Thanks again. You take care.
Great. You too. Bye-bye.